In the following experiment, we'll compare a sample of DNA collected from a crime scene to samples from 2 suspects. Label 3 microtest tubes, one for the crime scene sample and one for each suspect. Using a fresh micropipette for each transfer, add enzyme and a reaction buffer to each DNA sample. Cap the reaction tubes, tap gently to mix them, and allow the contents to accumulate at the bottom of the tubes. Incubate the tubes at 37 degrees for at least 30 minutes. When the samples are ready, they'll be loaded into an electrophoresis gel to compare the 3 samples. A gel tray is prepared with DNA samples placed in wells along one side. DNA molecules have a strong negative charge, so if an electric field is applied to the tray, they'll move toward the positive electrode. Agarose gels have microscopic pores, which act as a filter when molecules attempt to move through them. Smaller molecules will move through the gel more quickly than larger molecules. Those closest to the positive electrode are the smallest, and those farthest away are the largest. To prepare the gel, combine buffer, water, and agarose powder in a flask. Swirl to disperse the clumps. Boil the mixture to dissolve the powder, stirring occasionally. It should become clear like water. Cool the solution to 55 degrees and add distilled water to compensate for any evaporation that may have occurred. To prepare the gel bed, first close off the open ends of the gel bed using rubber dams. Make sure that the bed is clean and dry. Place a well former template, known as a comb, in the first set of notches nearest the end of the bed. Make sure the gel bed is on a level surface and pour the rest of the solution into it. Allow 20 minutes for it to solidify. It will be firm and cool to the touch. Once the gel is solidified, carefully and slowly remove the rubber dams. Then remove the comb by carefully pulling it straight up to avoid tearing the sample wells. Place the gel bed into the electrophoresis chamber, centered and level on the platform. Fill the chamber with diluted buffer. This type of gel is often called a submarine gel because it's submerged under a buffer. Add 5 microliters of gel loading solution. This solution contains glycerol to help the sample sink into the well, dye to help track the progression of the electrophoresis, and ethidium bromide, which glows when transilluminated. When loading a sample into a well, be careful not to tear the gel. Snap down the covers of the positive and negative electrode terminals, and make sure they're properly oriented. Connect them to the power source, and run the electrophoresis for a time specified by your instructor. The time required will vary depending on the voltage used, but should be at least 30 minutes. Bubbles will form on the electrodes if the current is flowing properly. Allow the tracking dye to migrate at least 4 centimeters to allow the DNA bands to separate. When it's ready, take it to a transilluminator to view the bands. When struck with the ultraviolet light of the transilluminator, the dye in the samples will luminesce. Since the enzymes split each person's DNA into a unique combination of lengths, the bands formed in the gels will be different for each person. If 2 samples result in the same pattern of bands, you know that they come from the same person.
For five from the album La Vie Chill by 4 Stones from Magnatune.com.